In this video, we'll continue talking about translation and finally get to how the genetic code, that is the sequence of DNA bases, is translated to sequences of amino acids, which determine the functions of proteins. So, recall that we left with transcription, which gave us a strand of mRNA, and that can be seen here. And now for translation, we really need to find the great kind of converter between the language of nucleotides, of bases, into the language of amino acids. And so this is a good time to introduce tRNA. So this is tRNA. tRNA, like mRNA, is made out of RNA. tRNA stands for transfer RNA because it actually is what's responsible for bringing in the amino acids. So the cool thing about tRNA is that as a tRNA molecule, it has the ability to form complementary bonds with mRNA. And so you can see that in mRNA here we have a codon, and a codon is just a series of 3 nucleotides. And on tRNA there's a section called the anticodon, which is the section of the tRNA that can bond to codons. And you'll see that here if the mRNA codon is GCC, the anticodon that will bind to that is CGG. And that again is the kind of complementary base pairing in RNA. The cool thing about tRNA is not only does it have this anticodon where it can interface with mRNA, it also brings along with it amino acids. And tRNAs with specific anticodons have specific amino acids that they bring. And so here is the kind of the big table here. And this is the table of the genetic code. It's how to go from bases, right, in mRNA into amino acids. And so the way you would read this is, so for instance, if we have the codon GCC in our mRNA, we go GCC, and that gives us ALA, which is the name of an amino acid. So this tRNA would bring ALA with it. And this specificity between the anticodon and the codon means that the sequence of mRNA can correspond directly to specific amino acids. And that's seen again in this table of the genetic code. And so translation occurs in ribosomes. And in those ribosomes, you basically get tRNA that comes in, and the tRNA with the anticodon that can bind to the codon of the mRNA comes in, drops off its amino acid. The amino acids bond, they form peptide bonds, and they all kind of shift over, and the next one comes in. And as this process continues, you're directly translating mRNA into amino acids. Another look at this. Again, so we have this as our ribosome. We have our strand of mRNA. And tRNAs will come, bring in amino acids, and when the anticodon lines up with the codon, they drop their amino acid off. Then a peptide bond forms between the amino acids already present and the new one, and everything slides over one, and you rinse and repeat. And basically what you end up doing is translating that mRNA, kind of codon by codon, into tRNA. And so now we can also see that depending on the mRNA sequence, you'll get different sequences of amino acid. But remember that this mRNA sequence came from the specific sequence of the DNA. So the specific sequence of the DNA bases creates a specific sequence for the mRNA bases. That mRNA is then broken down into codons, which are 3 nucleotides each, and these sets of 3, and each set of 3 corresponds to a specific tRNA that brings a specific amino acid. And that ordering of bases in the mRNA, which came from the DNA, dictates the ordering of amino acids that make up the protein, which, like we said before, determines the shape of the protein and ultimately what the protein can do.